Hi all, today we are discussing about cytokines and their properties. Cytokines, molecules that communicate among cells of the immune system are referred to as cytokines. In general, cytokines are soluble molecules, although some also exist in membrane-bound forms. The interaction of cytokine with its receptor on a target cell can cause changes in the expression of adhesion molecules and chemokine receptors on the target membrane, thus allowing it to move from one location to another. Cytokine can also signal an immune cell to increase or decrease the activity of particular enzymes or to change its transcriptional program, thereby altering and enhancing its effective function. They can instruct a cell when to survive and when to die, although the term cytokine refers to all molecules that communicate among immune cells. Properties of cytokines. First one is cytokine mediate the activation, proliferation and differentiation of target cells. That is, cytokines regulate the intensity and duration of the immune response by stimulating or inhibiting the activation, proliferation and or differentiation of various cells by regulating the secretion of other cytokines or of antibodies, or in some cases by actually inducing programmed cell death in the target cell. Cytokines and their fully assembled receptors exhibit very high affinity for one another, with dissociation constant for cytokines and their receptors ranging from 108 to 1012. Cytokines exhibit the attributes, it is the main point, attributes of pleiotropy, redundancy, synergism, antagonism and cascade induction, which permit them to regulate cellular activity in a coordinated interactive way. Cytokine that induces different biological effects depending on the nature of the target cell is said to have a pleiotropic action, whereas two or more cytokines that mediate similar function are said to be redundant. Cytokine synergy occurs when the combined effect of two cytokines on cellular activity is greater than the additive effects of the individual cytokines. In some cases, the effects of one cytokine inhibit and antagonize the effect of another. Cascade induction occurs when the action of one cytokine on a target cell induces that cell to produce one or more additional cytokines. Second one is cytokines have numerous biological functions. Although a variety of cells can secrete cytokines that instruct the immune system, the principal producers are Th cell, dendritic cell and macrophages. Cytokines released from these cell types are capable of activating entire network of interacting cells. Among the numerous physiological responses that require cytokine involvement are the generation of cellular and humoral immune responses, the induction of the inflammatory responses, the regulation of hematopoiesis and wound healing. Third one is cytokine can elicit and support the activation of specific T cell subpopulation. Helper T cells can be classified into subpopulation, each of which is responsible for the support of a different set of immune function. For example, Th1 cells secrete cytokine that promote the differentiation and activity of macrophages and cytotoxic T cell, thus leading to a primarily cytotoxic immune responses in which cells have been infected with viruses and intracellular bacteria are recognized and destroyed. Like that, cytokine can elicit and support the activation of specific T cell subpopulations. Fourth one is cell activation may alter the expression of receptors and adhesion molecules. The ability of cytokine to activate most, if not all, members of particular immune cell subpopulation appears to conflict with the established specificity of the immune system. In order for a cell to respond to a signaling molecule, it must express receptors for that molecule, and responsiveness to a molecular signal can thus be controlled by signal receptor expression. Furthermore, activation induced changes in the adhesion molecules that are expressed on the cell membrane ensures that simulated cells migrate to and then remain in the location best suited to their function. T cell activation by antigen also upregulate the expression of the receptor for cytokine that provide proliferative signals such as IL-2 and also for differentiative cytokines such as IL-4. IL-4. Like this, cell activation may alter the expression of receptors and addition molecules. Fifth one is cytokines are concentrated between secreting and target cell. During the process of T cell activation by an antigen presenting dendritic cell or by B cell activation by a cognate T cell, the respective pairs of cells are held in close juxtaposition for many hours. Over that time period, the cells release cytokines that bind to relevant receptors on the partner cell surface without ever entering the general circulation. Furthermore, during this period of close cell-cell contact, the secretory apparatus of the stimulating cell is oriented so that the cytokines are released right at the region of the cell membrane that is in closest contact with the recipient cell. Sixth one is signaling through multiple receptors can fine-tune a cellular response. Cytokine and chemokine signaling in the immune response can be strikingly complex and occasionally redundant affair. Effector molecules such as cytokines can bind to more than one receptor and receptor can bind to more than one signaling molecule. Effector molecule signaling can also cooperate with signaling through antigen specific receptor. Signals received through more than one receptor must then be integrated at the level of the biological response with multiple pathways acting to tune up or tune down the expression of particular transcription factors or the activity of particular enzyme. These include some cytokines and from where it is secreted, and some families that includes some cytokines are included in these tables. And the important point is cytokine antagonist. A number of proteins that inhibit the biological activity of cytokines have been reported. These proteins act in one of two ways. Either they bind directly to a cytokine receptor but fail to activate the cell, thus blocking the active cytokine from binding, or they bind directly to the cytokine itself, inhibiting its ability to bind to the cognate receptors. These cytokine antagonists are found in blood streams and extracellular fluids. Example, IL-1RA. These are all about cytokines and their properties. Thank you.